Thank you for the opportunity to give this talk, which will be about some translational work where we've combined molecular profiling of liquid biopsies with machine learning to determine tissue of origin in cancer of unknown primary. So cancer of unknown primary, or CUP, is cancer that's spread, but where the standard diagnostics have failed to identify the primary tumour site. So 20% of patients with CUP are in this favourable subset where there's some indication of the primary tumour site, and this can open up type-specific therapies for them. But the majority are in the unfavourable subset where there's no indication of the primary site, and they just get standard chemotherapy and typically have poor outcomes. So if we're able to determine tissue of origin for these patients, this could improve their treatment options. And we're exploring a liquid biopsy approach for doing this. And in particular, we're looking at methylation of cell-free DNA, or cfDNA. So cfDNA is DNA fragments that are released from cells into the bloodstream following cell death. The majority has hematopoietic origins, but you can get higher amounts in cancer patients due to circulating DNA from the tumour, so-called ctDNA. And the proportion of ctDNA within the cfDNA, the tumour fraction, is typically low, but varies depending on cancer type and stage. And it's known that different tissue types and different cancer types have different patterns of DNA methylation. So this means that the DNA methylation patterns on cfDNA can act as a kind of postcode, allowing us to determine the tissue of origin of the cfDNA fragments. So we've developed, so we're using an assay that was being developed in-house at the National Biomarker Centre called T7MBD-seq, which does genome-wide cfDNA methylation profiling. It's a methylation enrichment approach, so it captures fragments of DNA that are methylated and then sequences those fragments. And then we've also developed a next flow pipeline for processing this data, and ultimately we end up with these genome-wide methylation profiles of read counts, where you get these peaks of reads in regions of the genome that are methylated. And then we can use those profiles for downstream analysis, for example, determining differentially methylated regions between different cancer types and developing classifiers. And we've also built an R package called MESA to facilitate this type of analysis. So we developed a classifier called CUPiD, or CUPiD, to determine tissue of origin from cfDNA methylation profiles. So we don't have a sufficient number of cfDNA samples from cfDNA to train a tissue of origin classifier, because you need them across many cancer types. So instead, we've used methylation array data, primarily from TCGA. So this is over 9,000 tumor tissue samples from across 29 different cancer types. And we've converted the methylation array values to equivalent T7MBD-seq read counts. And you can see on this UMAP that the samples split nicely by cancer type based on their methylation profiles. And we determine differentially methylated regions pairwise between those cancer types to use as features in our classifier. So these are tumor tissue samples, so they look very different to cfDNA samples that have low tumor fraction. So we perform data augmentation to generate a training data set that mimics cfDNA. And to do this, we take reads from a non-cancer control, so a healthy cfDNA sample that we've sequenced, and we mix with that some reads from a tumor sample. And we repeat this lots of times with different combinations of samples, and also varying the proportion of tumor reads that are mixed in each time to mimic different tumor fractions. And this ultimately generates a large augmented training data set of these in silico mixture samples that we use along with our differentially methylated regions to train an ensemble of 100 extreme gradient boosting classifiers. So each classifier uses 80% of the data to train. Each has 29 tumor classes and one non-cancer class. And then we can apply those trained classifiers to a new cfDNA sample to get prediction scores for each class. And then if the average score for a class is above a 0.5 threshold, then that class is called as the tissue of origin. Otherwise, CUPiD returns an unclassified outcome. So we've applied CUPiD to 143 cfDNA samples from patients with metastatic cancer of known primary type. This is across 13 different types. And of those 143, CUPiD's predicted the correct cancer type for 121 of them, so that's 85% sensitivity. 18 samples were unclassified by CUPiD, the majority of these having low tumour fraction. So of the 125 that received a classification, 97% were correct. So that's on patients with known tumour type. We've also applied CUPiD to patients with CUP, to 41 patients, and CUPiD made a prediction for 32 of them. So 26 of these patients eventually did have a resolved or suspected primary diagnosis. And our CUPiD predictions aligned with these diagnoses or suspicions 88% of the time. But CUPiD could have facilitated the time to diagnosis. And then CUPiD was also able to make predictions for 6 out of 8 patients where there was no clinical suspicion. So that's obviously a very brief summary, but you can find out more details in our CUPiD paper. So we've shown that combining machine learning with cfDNA methylation profiling has the potential clinical utility for tissue of origin classification in CUP. But we have only tested it so far on 13 known tumour types, and we do see poorer performance in samples with lower tumour content and for certain classes. So we are continuing to optimise CUPiD in various different ways, so including improving our feature selection and our data curation, and we're also now starting to explore using generative AI approaches to improve our data augmentation. And we've got a PhD student, Holly Castle, who's working on some of these aspects, and please do check out her poster she's presenting tomorrow. So we'll go on to then further validate CUPiD in a larger set of known cancer types with a view to biomarker qualification in a prospective clinical trial, to hopefully get CUPiD available in the clinic for the benefit of patients. So this has been very much a multidisciplinary team effort. So I'll just pick out a few people. So Simon Pierce did a lot of the bioinformatics work, as a previous colleague, the experimental, and it was led on the clinical side by Alicia Conway and Natalie Cook. Thank you for your attention. Happy to take any questions.
Thank you, Stephen. Great talk. We've got time for one question.
Thank you. Thank you. Very nice talk. Just wondering, or maybe I missed it, that did you see any correlation of those methylation patterns in circulating free with the tissue methylation patterns? Was there any correlation that you see some kind similar in the tissue also?
Could you just repeat that? Sorry, it's a bit of...
You saw these methylation patterns in the circulating free DNA. Did you see that also in the biopsies at the tissue level? Correlation.
So these patients with CUP, so some of them have biopsies, but a lot of them don't have tissue biopsies. So I think the biopsy failure rate is very high, could be as high as 60%. But we do know that the methylation patterns do correlate very well in the cfDNA and tissue. We've seen that across lots of different studies.
Thank you, Stephen.